Hey Team Bio, welcome to your screencast on diversity created in Meiosis. As you recall from today's little lecture about sexual and asexual cell replication or reproduction, sexual and asexual reproduction, that diversity is the big benefit of sexual reproduction as it creates unique organisms that in a rapidly changing environment might have the combination of alleles that make them successful within that environment. So the screencast today is focusing in on how sexual reproduction or meiosis leads to diversity and the steps at which the most diversity is created. Okay, but quick, to go over a little bit of terminology, I want to start talking about the cell that is going to replicate here or divide, some of the terminology around this cell. Okay, so a chromosome, you'll see here this number 2n. This chromosome is the total number, 2 times n is the total number of chromosomes in what's called a diploid, diploid, diploid cell, where n is equal to the number of homologous chromosomes. So as you recall from the screencast yesterday, we have 23 pairs of homologous chromosomes, one from our biological mom, one from our biological dad for each set of chromosomes. So the total number of chromosomes that we have is 46. So our human N is equal to 23. We have 23 homologous chromosomes, and our human diploid cells have 46 chromosomes total. This cell has 4 chromosomes total, so its N is 2. It has 2 pairs of homologous chromosomes. So just a quick reminder, homologous chromosomes share all the same genes, but they have different alleles, different variants of those genes on those chromosomes. Okay, so in mitosis, we start with a diploid cell with 2n. If this is a human cell, it would have 46 chromosomes. And then at the end of mitosis, we are again left with 2 diploid cells, each with 46 chromosomes if this were a human, and their clones. So these 2 cells are identical, and they have the same number of chromosomes as the original cell. And this is true of what we call all somatic cells. Somatic cells are cells of the body which is in contrast to gametes, which are sex cells, which are eggs and sperm. So all of your cells, with the exception of your gametes, are somatic cells, and they're all identical. They all have 46 chromosomes, and they are all diploid. Okay, this is in contrast to the process of meiosis, which one of the goals of meiosis, including to create diversity, is to have the number of chromosomes to create what we call haploid cells. So the goal of meiosis, or one of the goals of meiosis, is to have the amount of genetic information. So here you see that all of these cells are diploid, they're 2n, and all the cells that we see down here are just n. So in this case, their homologous chromosome number is 2, so their total number of chromosomes in each cell is 2. But if this were a human egg or sperm, we would see 23 chromosomes in each of these cells. So we create 4 haploid cells. Oh, I guess I can also say we create 2 cells here, but this process creates 4 haploid cells with a unique scramble of genes. And I should say alleles because they don't actually have unique genes. They all should have the same genes because they're all the same species. Okay, so how do we get this unique scramble of alleles in the resulting gametes? That's what we're going to focus on today. Okay, so the unique scrambling begins during prophase 1 of meiosis. Remember, meiosis is going to have 2 cell divisions in order to create 4 cells at the end, whereas mitosis just has 1 cell division, which leads to 2 cells created at the end. Okay, so during prophase 1 of meiosis, we have this exchange of genetic information across homologous chromosomes that scrambles up the information that these chromosomes contain. So let's talk about how that happens. Homologous chromosomes, instead of chromosomes, all the chromosomes aligning on the metaphase plate, in prophase 1 of meiosis, homologous chromosomes line up next to one another. And recall that if this was the chromosome this organism got from its biological dad, then this would be the homologous chromosome of the pair that it got from its biological mother. These chromosomes are similar in that the homologous chromosomes contain all the same genes, but they have different alleles. Imagine this chromosome has a variation for hemoglobin. So the hemoglobin gene is located on both of these chromosomes, but there are different variations of the hemoglobin gene on these 2 chromosomes. So they're going to contain the same genes. They have the same variations, or the same, they have the same genes, is really the word for it, at the same spots, but different variations of those genes. So during prophase, there's this event called crossing over, where the genetic information or the 2 chromosomes align with one another, and they actually form a double-stranded break in each of these chromosomes. Because remember, a chromosome is just a long coil of DNA. Where they're going to form this break is called a chiasmata, or chiasma. What's going to happen is that a little piece of the chromosome that was originally on the homologous chromosome received from the mother is now on the chromosome that the father has, and vice versa. So we've exchanged little sections of these homologous chromosomes. So now these 2 recombinant chromatids have a unique combo of alleles. This didn't create any new alleles in the process. Well, maybe it could have, depending on where the break happened. But imagine you have 2 genes on here, the gene for hemoglobin and the gene for, sense of smell. And the 2 homologous chromosomes contain different variations. Now you've created a new chromosome here that has mixed up the pairing of the hemoglobin gene with the sense of smell gene. So we haven't created any new alleles here, but we have mixed up which alleles are pairing with other alleles on this chromosome. So this is a totally unique combination. And this crossing over event can take place on any location on the chromosome. And every meiosis is going to have lead to a different crossing over event. And so we're going to see different lengths of red and blue in any other meiosis process. So this, and we can have more than one crossing over event on every chromosome. So instead of it being like a long stretch of red and blue, it might look like a patchwork of red and blue along this chromatid and this chromatid. Okay, so this crossing over introduces an enormous amount of diversity within the chromosomes through this recombination. Okay, so that's one. This is the first way that meiosis is creating diversity through crossing over and the creation of recombinant chromatids. Okay, the second way diversity is created is this concept called independent assortment. Okay. The way that independent assortment works is that when these homologous chromosomes align on the metaphase plate, it is random which chromosome ends up on which side of the metaphase plate. So if you look at these two possibilities, in this possibility, we have both the chromosomes from the biological mother aligning on this side of the metaphase plate and both the chromosomes from the biological father aligning on this side of the metaphase plate. If this arrangement happens at metaphase 1 of meiosis, then we'll end up with two different combinations of cells. The big A alleles end up with the big B alleles in the gametes, and the little a alleles end up with the little b alleles in the gametes. So this is one possible alignment, which leads to two different types of gametes. Another equally possible or probable alignment is to have the big chromosome, chromosome 1 from mom on this side, and chromosome 2 from dad on this side of the metaphase plate. This alignment of the chromosomes leads to the big A allele ending up with the little b allele in this set of gametes, and the little a allele ending up with the big B allele in this set of gametes. So this is four possible combinations of alleles depending on the alignment of the homologous chromosomes on the metaphase plate. Okay, so this cell has two chromosomes. And four possible combinations. Of those chromosomes, depending on how they line up on the metaphase plate in metaphase 1 of meiosis. So there's actually a formula for determining the number of possibilities that independent assortment can lead to based on, this formula, which is 2 to the n, where remember n is equal to the number of homologous chromosomes. So in this case, our n is 2. We have two pairs of homologous chromosomes, so 2 squared is equal to 4, and as you see here, there are four possible combinations. So if you have only two pairs of homologous chromosomes, there are four possible combinations in this process of independent assortment. However, humans have 23 pairs of homologous chromosomes. So Rn is 23. 2 to the 23rd is a much bigger number. This is equal to 388,608 possibilities. So just the way the chromosomes are aligning on the metaphase plate can lead to over 8 million different combinations within the gametes. So this is also a huge amount of diversity created in this step of meiosis. So in combination with crossing over, we make this number even much bigger because now we have, so crossing over plus independent assortment leads to an enormous amount of diversity. Okay, the last bit of diversity that is introduced in this process is actually when two gametes meet. The end result of meiosis is 4 unique haploid cells, 4 unique gametes. So when this unique combination of alleles combines with another gamete, we get a unique organism. And I guess I should say this combining is during fertilization. Okay, so if this was the second way diversity is created in meiosis, then fertilization is the third way diversity is created, where we take one unique scramble of alleles and we're going to combine it with another unique scramble of alleles as an egg and a sperm fuse during fertilization, and we're going to end up with a totally unique organism. And there's another, oops. Okay. I think that is it for this screencast. I will see you all in class on Wednesday.